So with that, I would like to introduce Dr. Gustavo Diaz, a research scientist in the group, who will tell us today about proteomics and its microbial community extension of metaproteomics. Who is doing what in microbial communities? Thanks, Gustavo.
Thanks, Corey. Please let me know if you can hear me.
Yes.
Okay. So as Corey said, I will try to go over metaproteomics and see if we can answer this question with this type of analysis. Who is doing what in microbial communities? So I'm going to go over an overview of microbiome and metaproteomics. And then we're going to see the workflows that we use to do metaproteomics studies, analysis, and the challenges of each step. And finally, I will be just sharing with you a couple of examples of projects that we conduct here in the ARC metaproteomics projects. So let's start talking about a little bit of the microbiome as the subject of a study of metaproteomics. And as you know, the microbiome is a community of microorganisms that exists in a particular environment. In the particular case of the humans, the community of organisms living in a particular anatomical localization, such as the gut microbiome, the skin microbiome. So every surface of our body that is exposed to the environment is going to be covered by microorganisms. And I just want to show some numbers to give you a ground perspective of the relevance of the microbiome. So here in the picture, you will see in the figure in the left side, our body and the cells in our body, which are about 10 to the 14th in numbers. And we have, as you know, 23,000 genes. In the next part, in the middle, we'll see the invisible microbiome. And the numbers are just huge. So we have 1 million times more cells and around 500 times more genes. So the complete human is our cells plus our microbiome, and this is a normal functional body. And as you might think, the variations in this microbiome has important impact in our health. So just to show one of the examples of the relationship between microbiome and human health and disease is the case of atherosclerotic cardiovascular disease. It's a well-studied example of the relationship of the microbiome and our human health. And this slide that I'm presenting here is from a work recently published for one group, for the group of Dr. Whiton here at CSU. And what we are seeing here is like when we ingest diet with highly content of proteins, we end up having a bunch of in our intestines a lot of quaternary amines. These amines are primarily metabolized by our gut microbiome, and they will produce a compound, TMA, a compound that is exclusively produced by bacteria. And this TMA is going to get the portal circulation, get to the liver, and is going to be oxidized to TMA N oxide. And this TMA N oxide has been highly associated with ACVD. So as you might see in the center, if our microbiome, if the gut microbiome of a person is highly enriched by microorganisms with the enzymes that are able to produce TMA, so we will have a proteogenic microbiome. And on the other hand, if the organisms there are mostly producers of the MTXB, which is a demethylase, is going to do a demethylation of those quaternary amines, and then we are not going to produce that much TMA, and then it's going to be a protective microbiome. So there are some other examples where we can associate gut microbiome or microbiome with different physiological processes. And this one is, for me, is less intuitive, is how variation in the gut microbiome can cause or can be linked to mental disorders. And again, this is mostly related to metabolites that are produced by different bacteria in our guts that then end up circulating and causing effects, even in our brain. So the evidence is that strong that if you see here in the slide, there are so many clinical trials right now using probiotics, which are living bacteria, to modify microbiota in order to treat some mental conditions. So from the environmental perspective, I brought here this example of the soil. And it's just to show you some numbers of how many microorganisms do we have in a gram of soil. And you see, so the numbers are impressively high, and it is very easy to think how much of what will be the impact having this much of living organisms in the soil. So most of the information that we have about the microbiome and which is currently available is from Metagenomics. So Metagenomics, in a very general way of explaining, focus on the study of those that are known as marker gene studies, 16 sRNA to study bacteria, 18 sRNA for humans, and internal transcribed spacers when we want to study fungi. Or we can do whole genome shotgun metagenomics in order to get information from microbiome samples. And just generally speaking, we get reads from fragments of the genes that we obtain from the sample, and then we do some assemblies in a process now to build context. Those contexts are then put together in a process known as binning in order to gathering them around similar taxa. And finally, we can perform functional annotation and taxonomic profiling. So in general, what we can get from metagenomics is a survey of all genomes in an environmental sample or a human-related microbiome sample, and an idea of the functional capability. But what if we want to go beyond the genetic potential? What if we want to know what is actually happening? So I think that based on this quote from Professor Josh Olabayer, in which he explained in a clear way how proteins are the actions in biology, and also how they are key participants in the structure of all of living organisms. Maybe we need to start just thinking in proteins. So is how we start thinking in metaproteomics. And this is the definition that was given exactly 20 years ago. The large-scale characterization of the entire protein complement of environmental microbiota at a given time point. And studying the metaproteomics allow us to have a direct detection of the phenotype. The proteins are there, so we know that the phenotype is happening. And it's showing us what are those microorganisms producing in order to adapt to a specific environment or stimulus. And metaproteomics are also a perfect linkage between the genetic potential and the final products that we can get from metabolomics studies. So having the proteins is going to complete the whole picture of our study microbiomes. So to do this type of studies, this is a general overview of the workflow that we follow. Is in principle very similar to what we normally conduct to do proteomics or regular proteomics or single organism proteomics. But there are some particular challenges at each stage that I'm going to discuss in the following slides. So normally we get our sample. I say here bacterial extraction, but you know that the microbiome is also fungi and some eukaryotic parasites, or commensals, let's say, protein extraction, and then LC separation, mass spectrometry analysis, and then database search. you want to do a little bit more, want to see a little bit more of detail. So I just want to highlight that, for example, in the blue part related to sample processing and protein extraction, when we are dealing with metaproteomes or microbiomes, it's really key to take into account that we have there gram-positive bacteria, gram-negative bacteria, fungi, and in the case of soil, we have these humic compounds, and all of that is going to cause problems and challenges for the analysis. And then there are several options for the cleanup and separation. Separation is key because, you know, like, for example, when we work with proteomics, the complexity of the proteome makes super difficult the analysis, so we need to do some fractionation. And that's from, let's say, human or bovine. But if we have metaproteomics, we are talking about 3,000 or 5,000 different proteomes. So the orders of magnitude of the different concentration of proteins is huge. So it's going to be important to consider early on in the preparation of an experiment some sort of fractionation separation. And finally, bioinformatics, the analysis part, is very challenging, and I will discuss that in further slides. So generally speaking, we use for metaproteomics something known as shotgun proteomics. It's very similar as we do when we do single proteomic studies. And in shotgun proteomics, what we want is to put the proteins into peptides. So we denature the protein, we reduce those cysteine bridges, and then we alkylate them so they don't form again. That will create a modification in the mass of the cysteines of 57 Daltons. And then we use endoproteinase such as trypsin to cut the protein into peptides. Trypsin, for example, cuts our protein every time there is a lysine or an arginine. In those letters there, the amino acid chain is the K or R. You will see how the cuts are flanking those two amino acids. And then we are able to get this solution of peptides that is going to be injected into the LC to be separated. It's a little tiny fraction of the sample. It's just 1 microgram of peptides that is injected in the liquid chromatography column. And then the sample is separated. Those peptides are separated by the hydrophobicity. And then they are generally coupled to an ionization source. And the ionized peptides are first analyzed with one cycle of mass spectrometry, and some of those are going to be selected to be fragmented to produce MS spectra. Here in the figure, this is an example of the chromatogram, and we have all those peptides across the retention time in the analytical column. So at the end, what we get experimentally is MS1, thousands of MS1 spectrats, much more of MS2 spectra. But we need to provide to researchers meaningful information, which are list of proteins with some number of relative quantification. So what we do to provide this information is using informatics, and we rely on protein sequences that are already predicted from genome sequences. They are normally in repositories. One of the most used ones is the UniProt repository. And what we do is, like, using in silico tools, we conduct the exact same procedure as we did with our experimental samples in silico. So we get we cleave the amino acid sequence using a protease, and we get all the theoretical fragments from the sequence, getting the information from these similar to what we get in the MS1. And then, since the fragmentation of the peptides is also predictable, we will get all the theoretical possibilities for the MS2. Again, using informatics, we compare the experimental MS2 with the actual MSMS that we get from our samples. Since the distance that we got, this is an example here, since the distance that we have in an MS2 from one peak to the other peak is equal to the mass of an amino acid, we're able to predict the amino acid sequence. And then the amino acid sequence is rolled up into a protein, and the protein to the actual gene that will give us information related to function and taxonomy. So since this process relies on algorithms to do these matches, we need to test the accuracy of this matching. And one of the well-established methods to control this is the target decoy control for discovery rate. So we want to see if this prediction is accurate. So what we do is to create a decoy data set, which is our forward data set. What we do is scramble that or produce them in a reverse way, so in a way that makes no sense. It shouldn't be any hits. If we search our actual MSMS using that data set, in theory it should be 0, but it's not. And depending on the quality of our MSMS spectra, and also the size of these data sets, is going to be the quality of our analysis. So when we are studying single organisms, it's relatively manageable. So I want to, here you see an example of how many theoretical sequences we have from Bacillus subtilis. We have around 4,000 sequences, and with the decoy, it's going to be 8,000 sequences. But what happened when we have these massive metaproteomics data sets? So in the right side, we have these theoretical sequences from the whole prokaryotes, and it's more than 36 million sequences. So with the decoy, it's going to be 72 million sequences. So, and that's one thing. The other part is, even though there is that many information available, there's going to be a bunch of microorganisms that we don't have any information about them. So it's going to be missing information for several of them. Then the large number of entries that we have to study is going to challenge any matching algorithm that we use. And obviously, the search space being so big, the decoy data set being that big, is going to impact our fall discoveries. So that takes us to the first big challenge that we have to do metaproteomics studies. And one of the possible solutions is searching subsets of large databases that will increase our identifications. So one of our close collaborators, Professor Jagtap, introduced a method known as the iterative database search, and it's a two-step interrogation strategy. And what he did was, or his group did this initial search using a whole pan-metaproteome, using reference genomes and metagenomic data, and without any FDR filtering, no fall discovery rate filtering, and using all the hits that they get from the first search, they built a second data set that was smaller. And with that small data set, they conduct a second search analysis that in which they use a highly stringent fall discovery rate. So in this publication, they from Proteomics 2015, the figure in the right side is showing us several comparisons of peptides identifications that are impacted by the size of the database that we use. The first one is the human intestinal metaproteome database. It contains bacterial, metagenomes, and human information, and it has over 6 million entries. The second one is a subset of 124 individuals, the intestine metagenomes of 124 individuals. It only has above 3 million entries. The third one is the genomes of 594 bacterial genomes that are commonly found in our guts, in our gut microbiome. It's 1.8 million entries. And the last one is using the human intestine metaproteome to conduct the two-step approach developed by Dr. Jagtap. And the second data set that was obtained to do the comprehensive analysis is less than 1% of the size of the original one. It's 90,000 entries. And if you see the figure, the number of the identification is significantly higher when we use the two-step approach. So another approach to create these subsets of large databases to increase the number of identifications in metaproteomics studies is based on the rationale is that if a microorganism is present in my microbiome, it is very likely that we are able to find highly abundant proteins in the sample. Those highly abundant proteins are ribosomal proteins and elongation factors. So in this case, the first search is known as the profile metaproteomics, profile the metaproteome. And what we do is like using all reference genomes, all genomic information, and we extract from that highly abundant proteins sequences. And we build this highly abundant protein database. Then we do a search using our experimental data at a false discovery rate of 1%. And we are able to identify taxa associated with the sample. And then selecting all the proteomes of any of the organisms linked to the high abundant protein that we found in the profiling search, we are going to build an expanded database that is going to be more or less customized for our sample set. This strategy has also been tested. Actually, this is the one that we prefer here in the ARC to conduct our metaproteomics analysis. Well, at this level here, what we get is a list of proteins that are present in our sample. But still, we need to know these proteins are linked to which taxa. And there is a second major challenge in metaproteomics, how we get this association entre peptide and taxa. And here we face this problem that is the peptide degeneracy, which is that a peptide can match several proteins. That can even happen when we do single proteomics analysis. We have a peptide in the left, and it can perfectly matches dozens of proteins. At the same time, we can link that several proteins to different taxa. So to solve this, in a nice approach was developed by a platform known as UniPet, in which the taxa associated with all matched proteins are then summarized as the lowest common ancestor, which is the most specific node in the taxonomic tree. That is a pairing of all matched taxa. And this allows us to centralize the information and obtain from a particular peptide a single assignment at the whatever lowest common ancestor we get. If the lowest common ancestor is phylum, that peptide is less informative for our purpose here, because a phylum will be connected to thousands of potential taxa. However, we get several assignments in which the LCA is a genus and species. So we use this tool. It's known as a UniPet, and to streamline the process, what they did was to generate all the peptides and all the assignments to its LCA for all the peptides available in UniProt. Then they store this information in a server, and we will be able to just use those databases to search our peptides from our sample sets. And here we have now a taxonomic assignment of our peptides. Normally we get information that is very similar to what is obtained with metagenomics in terms of taxonomic assignments. But again, what we want to know is what are those microorganisms doing? So what are the functions, the biological functions? And that's the true potential of metaproteomics. So that takes us to the third major challenge in metaproteomics analysis, and is the peptide function assignment. So, and again, here I just brought the example of UniPet because it's a very strong strategy. So here we have again the problem of assignment because, as I just told you, a peptide can be linked to several proteins. Because of that, a peptide can be linked to several biological processes, several biological functions. So it seems like we are not going to be able to connect a peptide with a single biological function. But what we can do is to summarize all the biological functions that are potentially linked to a particular peptide. So in the figure here, what we have is 3 different proteins, and they are connected to different biological functions that are depicted in those labels: 1, 2, 3 for protein A, 2 and 3 for protein 2, and 2, 5, and 6 for protein 3. If we digest the protein, every single peptide from each of the proteins is going to inherit their biological function. But as you know, protein 1, 2, and 3 can share a peptide. And in the example, they do. So the peptide that is surrounded by those dashed red lines is the same for the T proteins. So if we are analyzing that sample, so what we do is like we get the functional annotations or all the annotations that are linked to that peptide are summarized depending on the number of proteins that are shared. So for example, if you see the peptide present in the sample that is shared is linked to the Go term number 1, and it's shared for 3 proteins. So it's going to have let's say, an average participation of the functional capacity of that peptide in 1/3, whereas the Go term number 2 is in the 3 proteins. So it's going to have an assignment of 3 out of 3, and so on and so forth. When we have our analysis and we get in our experimental sample that particular peptide, what we do is like if we can assign to our sample all of those Go terms, all of those biological functions. On top of that, what we do is like we use the information, the abundance information for our proteomics analysis to give us a quantitative participation of that particular function in the sample. So again, what they do to streamline this process is to use all available tryptic peptides in UniProt in order to generate a pre-assignment of peptides to biological functions, and also to enzyme commission the EC numbers. So we have not only Go terms that are biological function, biological localization, but also enzyme commission. So those are basically all the processes that we need to face in order to conduct a complete metaproteomic study. As you might see, the majority of the challenges are based on the analysis part, the bioinformatic part. Fortunately for all of us, we have nice strategies to tackle those, and still some of them are still in development, especially the functional annotation part is difficult because of the shared information among peptides and proteins and among all the taxa. So now what I'm going to show you is what we have here at CSU to conduct this type of analysis. So this is the instrumentation that we have. This was funded for us to improve our capacity to do metaproteomics, and it's a collection of instruments, a liquid chromatography, Vanquish Neo in the left, excuse me, and a thermal orbit trap eclipse, a mass spectrometer, or it's a tribid. It has 3 mass analyzers in a single instrument, And we have this famous duo pro interface, which is a orthogonal, it allows us for an orthogonal separation relative to both the LC and the MS. And it's especially useful for highly complex samples, such as the metaproteomic samples. So now what I'm going to share with you is a couple of examples of projects that we have been actively working here in the ARC Metaproteomics projects. So the first one is these changes in the gut metaproteome of women with different body mass index status. So we have this cohort of three groups of women with different mass index: normal, overweight, and obese. This is not a perfect metric for the to call these conditions, but it's what we have. And then we get a stool sample from them, and we extracted proteins from these stool samples following our one of the protocols that we have here. Then digested peptides were injected into our system, LC separation, and then gas fractionation with FAIMS before going to MSMS analysis. This project is a collaboration of the Department of Horticulture, Department of Soil and Crop Sciences, Thermoscientific, and the ARC. So far that we have been able to obtain is like a nice number of proteins. This is a huge number of proteins out of a metaproteomic study, more than 22,000 proteins and more than 90,000 peptides. And you see in the left side, there is a great deal of overlapping of the proteins that we found, but there are some interesting proteins that are particularly linked to each of the states. Obese, overweight, and normal. Over 1,500 proteins in each of the groups are proteins that are exclusively in those groups. When we did the taxonomic analysis using peptides, we were able to find a nice amount of species. We were able to get to the genus and species with interesting numbers, but more importantly, what we get was information of the most abundant phyla that were previously described in other studies. So with metagenomics and other metaproteomics studies, in which like Firmicutes, Bacteroidetes, and Actinomycetes are the most abundant phylums in our gut microbiome. And our preliminary metaproteomics analysis, looking for changes in metabolic activity, show us that the microbiome for normal individuals had a higher number for all the processes associated with glycolytic and energy production. And this is also based on our analysis of peptides. Another study that I'm going to mention is this method optimization study. One of the challenges studying metaproteomics, as I told you before, is the nature of the sample, and soil metaproteomics is very challenging, not only because of the variety of microorganisms, but because of the humic compounds. So we thoroughly analyzed two methods, and one with a strong mechanical approaches, and the other with strong chemical approaches. We also did different precipitation strategies, acetone and TCA, and for strong mechanical approaches, we use silica beads and cap horn sonicator. And we did the extraction of the proteins, digestion to peptides, and then we injected that and we processed with our instruments. So far, what we have is we were able to find that protein extraction methods selects for proteins that are significantly different, even using the exact same sample. Meaning, depending on how you extract the proteins from those microorganisms, or how you precipitate the proteins, you are kind of having a bias on the selection of the proteins. This is critical for analysis, and it might be critical to respond to biological questions. And here in the figure that we see is the fold change in the x-axis and in the y-axis, the p-value for the significance of the difference in this fold change. And in the red square, you see there are hundreds of proteins that are significantly increased, whereas in the green square, there are hundreds of proteins significantly decreased. And also, one of the great findings or major findings that we are having so far is that strong mechanical approaches, such as bit beating and cap horn sonicator, all of those used in method A, facilitated the extraction of proteins from gram-positive bacteria. As you know, gram-positive bacteria has this thick cell wall rich in peptidoglycan that normally make them stronger and more difficult to break apart. So I think that's all that I have for today. Before going to giving the space for some questions, we want to invite everyone for a Metaproteomics workshop that we are going to be conducting here in the ARC. It's going to be this summer, the week of July 8th to July 12th, and it's going to be like in presence. Also, we're going to have some remote sessions. The target audience: faculty, research scientists, postdocs, PhD students, and we have capacity for more or less 10 participants for hands-on experiments, and there will be wider space for lectures that will be given by remote strategies. So with that, we'll be open to some questions or comments.
Thank you, Gustavo. Questions from the audience?
Hey, Gustavo, it's Kitty.
Hi, Kitty.
Just curious, the study with the human gut microbiome, did you see, I mean, obviously you saw differences in kind of some of the pathways that might be involved. Did you see any like potentially interesting differences in the distribution of the microorganisms per group?
Yes. Yeah, we have seen some differences. In fact, I still need to do some additional analysis to get a full list of microorganisms based on metaproteomics analysis. But I was able to get at the level of seeing variation at the phylum level, linked to phylum is a big group, but linked to microorganisms that were in which contain those that are TMA producers, or TMA metabolizers. So that's something that was reassuring.
I see a hand raised from Say.
Hi, Gustavo. Do you hear me?
Yes.
Oh, yeah, wonderful. I have a question also regarding the human gut metaproteome, comparing the people with the obesity. And then I see that those statistics already corrected using the multiple sampling correction. And do you think those data need to be also corrected or normalized based on each individual having different diet or certain occasion like sickness or different diet during the sampling?
Absolutely, Say that's super critical, and the metadata and the full study. Hopefully, we will be able to get this we have this metadata. I know that general information of, for example, if the diet was rich in proteins, information like that, because like a BMI is such a general marker. So, but we have information on data. It's a lifestyle, those general surveys that will give us some other levels of, let's say, grouping, and those need to be considered for this type of analysis.
I see. And then for those kind of study what's the ideal number of patient or cohort in each group? If more is better.
More is better. Yeah, that's beyond my knowledge, but definitely 8 is that. I mean, 20 we have like a 24, 25. That's not a great number. It's just exploratory study to do a proof of concept. Like a Metaproteomics is able to give us information that is linked to previous and other studies that have been performed with metagenomics. And especially you see here, I didn't mention this, but it was female cohort. There's also this is kind of a lack of information in the obesity and females. So we want to have this as a proof of concept of the methods, also getting a little bit into the biology, but definitely to have a full study, we would require an N bigger than that.
I see. The reason I'm asking this is I guess it's quite challenging to get many patients when you need more. But in my case, I work on air biome, and then that number can be manageable. So I was wondering what number of the biorep I need to aim to get.
Yeah, and that's challenging. And I know we know your samples; those are very challenging. So,
Right.
yeah, but first, we need to start having the great numbers, and there might be a metagenomics information. That will be a great guide for us just to see like the entire variation from genomic information. So we're able to do focus searches, and see proteomics is giving more information about the function of those microorganisms up there. Yeah, but I think that would be a discussion for a biostatistician to help you guide those, those methodological questions. Yeah, we can certainly have some ideas here. So, but definitely it's great to put some statistician in the table.
Yeah, we can discuss further later. Thank you.
Say.
Other questions? See a lot of muted microphones. Okay, well, I think we'll stop here then. Obviously, if you do have questions for Gustavo, don't hesitate to reach out after the talk as well. Thank you, Gustavo, for the presentation. Just a quick reminder for everyone, our next seminar is March 6, 2024, and will be presented by Dr. Claudia Boot on self-service mass spectrometry. Thank you, everyone, for attending.